The C. elegans genes Z3, Z4, and Z9 can mutate to cause a general block in apoptosis. For this reason, they are components of the commitment step, during which cells irreversibly commit to the apoptotic fate. CES1 and CES2, which can mutate to specifically block the death of the NSM sister cells, act in the cell death specification step, the step before the commitment step. And Z1 and Z2, which can mutate to block the engulfment of apoptotic corpses, act in the last step, the step during which cells are dismantled, phagocytosed, and degraded. There is at least one more gene that can mutate to cause a general block in apoptosis, and that for this reason is a component of the central commitment step. This gene is AGO1, the gene that we have heard of already in the last two sections. Dominant mutations in the AGO1 gene were identified in screens for egg-laying defective mutants. A total of 7 such dominant mutations came out of these screens. All these mutations cause the H's in neurons, which in hermaphrodites are required for egg-laying, to inappropriately undergo programmed cell death. What could be the normal function of the AGO1 gene? Since all 7 mutations were dominant, this wasn't clear. Could it be that the AGO1 gene plays a role in sex determination, and that the dominant AGO1 mutations switch the sexual fate of the HSNs in hermaphrodites to that of males? Remember, the HSNs normally die in males. Or could it be that the AGO1 gene is another cell death specification gene, specifying the death of the HSNs? What was needed was a loss of function mutation of AGO1. And when I started my postdoc in Bob Horwitz's lab, that's what I wanted to search for. The approach I took was a reversion screen. But the AGO1 reversion screen was done a bit differently than the Z9 N1950 reversion screen. When I started to work on AGO1, 7 dominant AGO1 mutations had been isolated, and they all caused an AGO phenotype. However, some of these mutations were fully dominant, whereas others were what is referred to as semi-dominant. A mutation is called fully dominant if 100% of animals heterozygous for the mutation exhibit the mutant phenotype. And a mutation is called semi-dominant if less than 100% of animals heterozygous for the mutation exhibit the mutant phenotype. In the case of the AGO1 mutation in 1084, for example, 100% of animals that are homozygous for in 1084 are AGO, but only 71% of animals that are heterozygous for in 1084 are AGO. Interestingly, around 70% of animals heterozygous for in 1084 and a deficiency that deletes the AGO1 locus on chromosome 5 are AGO as well. What this means is that if one of the N1084 alleles in a homozygous N1084 strain is hit by a second mutation, and if this second mutation converts the allele into a loss of function mutation, the animal should have about a 30% chance of being non-AGO defective. So the screen I did was the following. I mutagenized homozygous N1084 hermaphrodites with EMS and screened their F1 progeny for rare non-AGO animals. I screened about 30,000 F1 animals and identified 7 animals that were non-AGO, which was more than I had expected. And later I found out that all 7 animals actually carried the same mutation, a 5 base pair deletion. This deletion was most likely not induced by EMS, but had arisen spontaneously in the AGO1 and 1084 population that I had used for the screen. As we will see later, the AGO1 transcription unit is very small, significantly smaller in size than the average C. elegans transcription unit. And for this reason, it provides not a good target for EMS because there are just not many GC base pairs that can mutate to have a significant detectable effect on the function of the AGO1 gene product. In retrospect, I probably would never have found what I was looking for using EMS as a mutant. So I was lucky that this spontaneous mutation had occurred. As I just mentioned, I found 7 F1 animals that were no longer egg-laying defective. These animals were heterozygous for the N1084 allele and the new modified N1084 allele that carries the second mutation. I homozygosed the allele referred to as N1084 and N3082 and analyzed the animals. Homozygous N1084 and N3082 animals looked like wild type, and they produced viable progeny. I furthermore found that the HSNs were present again in N1084 and N3082 hermaphrodites. So what does N3082, the second mutation, do? There were at least 3 possible explanations. First, N3082 might revert the N1084 allele back to the wild type allele. In that case, N1084 and N3082 animals should be completely wild type. Second, N3082 might specifically block the death of the HSNs. In that case, the death of the HSNs, but not other cells, should be blocked in both hermaphrodites and males. And third, N3082 might cause a general block in apoptosis. In that case, the death of the HSNs as well as the death of many of the 131 cells that normally die should be blocked. To find out, I analyzed the pharynx of N1084 and N3082 animals using DIC microscopy. And what I found was that there were on average 12 extra cells in the anterior pharynx. N3082 therefore causes a general block in apoptosis. N3082 also turns out to be recessive and therefore a loss of function mutation of the AGO1 gene. These results demonstrate that the AGO1 gene is required for apoptosis. These results also demonstrated that, like the genes Z3, Z4, and Z9, the AGO1 gene acts in the commitment step. There are 4 genes now acting in this step. 3 of them required for the apoptotic death of many of the 131 cells that normally die, and that are therefore also referred to as pro-apoptotic: Z3, Z4, and AGO1. And 1 gene whose normal function is to prevent apoptosis, and that is therefore referred to as an anti-apoptotic gene, Z9. The loss of the pro-apoptotic genes causes a general block in apoptosis, whereas the loss of the anti-apoptotic gene causes ectopic apoptosis, the opposite phenotype. What's the order in which these 4 genes function? Is there an order? These genes are clearly involved in the same process, the commitment to the apoptotic fate. And some of them have opposite functions, pro or anti-apoptotic functions. In addition, loss of function mutations are available and cause opposite phenotypes. What one can do to order gene function in this case is double mutant or epistasis analysis. What is epistasis? Epistasis is a form of gene interaction in which a mutation in gene A masks the phenotype caused by a mutation in gene B. In this case, gene A is said to be epistatic to gene B. And the implication of epistasis is a negative or antagonistic interaction of genes, in this case gene A and gene B, in some biochemical or developmental sequence. Double mutants were made between the genes that cause opposite phenotypes when inactivated. Double mutants were made basically carrying a Z9 loss of function mutation and either a loss of function mutation of Z3, Z4, or AGO1. In the double mutants, cell death was then analyzed. Remember, the loss of Z9 function results in ectopic apoptosis and therefore embryonic lethality. The Z9, Z3 double mutants were viable, and they had a general block in apoptosis, a Z phenotype. For this reason, the loss of Z3 function suppresses or masks the loss of Z9 function, and Z3 is therefore epistatic to Z9. The same was found with Z4. The Z9, Z4 double mutants are viable, and they have a Z phenotype. The Z4 gene is therefore epistatic to Z9. In the case of AGO1, however, the opposite was found. A Z9 AGO1 double is not viable, and it still exhibits a ectopic apoptosis phenotype. For this reason, Z9 is epistatic to AGO1. How does the knowledge of who is epistatic to whom help us to order gene function? The commitment step can be considered a regulatory pathway or cascade with two alternative states: apoptosis or no apoptosis, like an on-off switch. In that case, epistatic indicates downstream. Z4 and Z3 are epistatic to Z9. The ectopic apoptosis induced by the loss of Z9 is dependent on a functional Z3 gene and on a functional Z4 gene. Z3 and Z4 therefore are required or act downstream of Z9 to cause apoptosis. Furthermore, Z9 normally acts to block the activity of Z3 and Z4. In contrast, Z9 is epistatic to AGO1. The ectopic apoptosis induced by the loss of Z9 is not dependent on a functional AGO1 gene. AGO1 is therefore not required downstream of Z9 to cause apoptosis. Instead, it acts upstream of Z9 to block its anti-apoptotic function. Based on these genetic considerations, the functions of Z3, Z4, Z9, and AGO1 can be ordered in the following genetic pathway. This is a genetic pathway, and the interactions shown are between the genes' functions. These interactions do not necessarily reflect physical interactions between the products of these genes. One more note on epistasis and epistatic interactions. Remember that the first mutation in the Z3 gene, N717, was identified as a suppressor of the Z1 mutation E1735. The loss of Z3 masks the loss of Z1, which means that Z3 is epistatic to Z1. Based on what I just told you, that would mean that in a genetic pathway, Z3 acts downstream of Z1. However, I also told you that while Z3 acts in the central step of the apoptotic process, the step during which cells irreversibly commit to the apoptotic fate, Z1 acts in the last step, the step during which cells are dismantled, engulfed, and degraded. How can it be that Z3 is epistatic to Z1? The specification of which cells should live and die, the commitment of these cells to the apoptotic fate, and their subsequent dismantling, engulfment, and degradation are a sequence of cell biological and biochemical events, with one event being dependent on the successful completion of the previous one. A kind of precursor-product relationship exists between these different stages. For this reason, epistatic indicates upstream in this case. The way I look at it is that if the commitment step is defective and no cells ever commit to the apoptotic fate, then it doesn't matter whether engulfment works or not. In other words, when you are working with genes that are components of what can be considered a regulatory cascade with an on-off switch, such as the commitment step, then in a double mutant, you will see the phenotype caused by the genes that act closest to the actual switch. Epistatic therefore means downstream. In contrast, when you are working with genes that are components of different steps that act sequentially and that each are dependent on the completion of the previous step in a double mutant, you will see the phenotype caused by the gene that acts most upstream, since once blocked, the cascade is interrupted and cannot proceed to the next step. When you are performing epistasis analyses, you therefore should make sure that you know what kind of cascade or processes you are looking at. And one note, the term epistasis is nowadays used by quantitative geneticists in a somewhat different way. It can be used to describe any non-additive phenotypic effects seen in individuals with a specific genotype. Now coming back to the genetic pathway of the commitment step. The loss of SET3 or SET4 causes a general block in apoptosis, and they both act downstream of SET9. For this reason, the order of these two genes could not be resolved, at least not using loss of function mutations. Once the two genes were cloned and identified, they were individually overexpressed in cells in C. elegans that normally survive. And what was found is that both could induce ectopic cell death. This provided an assay to determine whether SET4-induced ectopic cell death was dependent on a functional SET3 gene, and vice versa, whether SET3-induced ectopic cell death was dependent on a functional SET4 gene. The result of these studies was that the overexpression of SET3 can induce apoptosis in the absence of SET4, whereas SET4 is unable to induce apoptosis in the absence of SET3. The conclusion from this result is that SET4 acts upstream of SET3 to activate SET3, and that SET3 activation can be sufficient to commit cells to the apoptotic fate to trigger the switch, basically. Here now, the genetic pathway of the commitment step. It is a simple linear pathway with 4 components. The interactions between these 4 components are either negative or antagonistic, as between AGO1 and SET9, or SET9 and SET4, or positive or activating, as between SET4 and SET3. Based on this pathway, one can predict the activities of the genes. For example, when the most upstream component of the pathway, AGO1, is active, then SET9 should be inactive, SET4 active, and SET3 active. This should lead to a commitment to the apoptotic fate and therefore death. Conversely, when AGO1 is inactive, then SET9 should be active, SET4 inactive, and also SET3 inactive. This should not lead to a commitment to the apoptotic fate.